Okay, so it's 2 o'clock Eastern right now, so I figure it might be a good time to get started. Thank you all so much for joining today. My name's Bill Wysocki. I am the lead for the user services team in the Genomic Data Commons. And today I'd like to talk to you about an overview of the Genomic Data Commons. Um, just in case some of you weren't here for the very beginning or just before the webinar, um, please put all questions and answers into the Q&A field. I think there's a chat field too, but put it in the Q&A field so we don't miss them. All questions will be answered at the end. And if we can't answer any questions, we encourage you to contact the GDC help desk, which we will talk about later in this presentation. Additionally, this will be recorded and be made available via the GDC website. We do need a little bit of time to make this available. It won't be made available right away, but it will be in a couple of weeks or so. The agenda for today is basically a run through most of the features that the GDC has to offer. We're going to go over a short introduction of what the GDC is, ways to access GDC data, the analysis, visualization, and exploration tools offered by the GDC, how data can be submitted to the GDC, the bioinformatics pipelines, and then last, we're going to go over some GDC features and changes that are upcoming. So, as usual, we'll give a short introduction to what the GDC is. And in short, it's a data repository for cancer genomics. In addition to the molecular data that many of you might be familiar with and download from the GDC, the GDC also offers biospecimen and clinical data associated with that molecular data. Um, all of this data is processed using a set of standardized pipelines and is stored in a flexible data model to make sure that data is retrievable and um, and robust so that it can be processed by researchers. The data that actually comes into the GDC is submitted by external researchers, many of which are NCI-sponsored projects, and this data, as I said before, is processed by bioinformatics pipelines, and the outgoing data, which is available usually as an alignment and as downstream processed files, are made available to the wider research community. Um, this next slide is just a short summary of what the GDC has to offer in terms of data. This is as of the 29th of March, 2021. We are currently on data release 28, and things are ever changing in the GDC. So if you're watching this in the future, there might be more data. So there's around 3.5 petabytes of data across around 615,000 data files. This includes both the active data that is harmonized by the GDC and the legacy data that was inherited by the GDC, most of which is TCGA, Cancer Genome Atlas. Almost 85,000 cases are in the GDC across 68 projects. And for those of you who aren't familiar, cases are what we refer to patients or donors. Usually they do correspond to patients. In addition to these files and cases, we have 3.4 million explorable somatic mutations that are available through our portals. The three main types of data that the GDC offers are molecular data, clinical data, and biospecimen data, like I mentioned before. The molecular data includes data from multiple experimental strategies, and this harmonized data starts at the alignment and includes downstream derived data. So things like transcriptome expression or variant calls are made available in addition to the full alignment. Some examples of the experimental strategies are here: whole exome, whole genome, RNA-seq, etc. The clinical data that is made available in the GDC is uploaded as metadata associated with the patients, but it's also available as supplement files. So this includes the demographic, the diagnosis, exposure, that kind of information. So usually anything that doesn't fit into the GDC data model, and this is usually is because it's too specialized or maybe too sparse, we may try to make everything available to users. So that kind of thing will be made available in a supplement file. Lastly, the biospecimen data is also uploaded as metadata in the same way that this the clinical data is made available as metadata through the API or as supplement files, the biospecimen data is made available in that way too. At the bottom here, I'm probably not going to mention this every time, but I want to make you aware that in the past we've done many of these monthly webinars, and a lot of the features that I talk about today were were talked about a lot more in depth in a past webinar. So on the bottom, you'll see some months. These are webinars that went more in depth about each of these features. And if you go to the GDC website webinar page, you can scroll down. I believe they are in reverse chronological order, and you can come upon whichever one you need to. So all data in the GDC adheres to the GDC data model. This is a graph model, and it's designed to be strict to maintain data integrity, but flexible to accommodate many different properties. Just a quick overview, we have the case, which kind of falls right in the middle, this blue node here. It's surrounded by these purple clinical nodes, and downstream are all of the various bioinformatics processing pipelines, as well as the biospecimen nodes or entities. Um, so because many different projects are submitting many different types of data to the GDC, we make changes to the data model about once every quarter. And this is based on requested changes from submission groups that are currently submitting and upcoming projects. And we'll usually determine, which fields are important enough to go in our API and which ones will end up in the supplement files. So the next thing I want to talk to you about are the GDC data access tools, how the GDC makes data available to the public. And the first data access tool I'd like to introduce you to is the GDC data portal. And real quick, I'm going to do a very short demo on what the GDC data portal has to offer. So here we are on the GDC data portal homepage, and what I want to go over is the repository. The repository is where the files that are harmonized, the 615,000 files, are available for users to download. So hypothetically, let's go over a query that some researcher might have. So let's say I'm a researcher. I know I want to use TCGA data. I would click that as the program. I'm interested in kidney cancer, so I will click on the primary site, kidney. And let's say I'm interested in only kidney cancer in males. I would click gender male. At this point, I have a cohort of 623 patients, and now I want to boil this down to the files that are made available, and I will do that using the file faceted search. So each of these facets corresponds to a different property of the file. In this case, I might be interested in experimental strategies. So maybe I'm interested in the whole exome sequencing. And let's say I know that I want to download the open access MAF files. I would click on Data Format, MAF, and for access, I would click Open, as you might expect. Now that I have a list of files, I have a file size. It's around 70 megabytes. I will add these files to the cart, go to the cart. I can see these 16 files here across 4 different projects, and I could download them directly through the portal. Here I downloaded the open access files. But if a user wanted to download controlled access files, they could do that by logging in. Whoops, that automatically opened. They could do that by logging in here and entering their ERA Commons ID. And that is how we retrieve which projects each user has dbGaP access to. So in order to download controlled data, you need dbGaP access to a particular project. Now back to the slides. I will skip through these since I went over that. Um, so for the more technically savvy people here, there is the GDC API. This is how to access data via the command line. This allows users to query, submit, or download data directly using the command line. Um, Many people access the GDC API using curl. That's what I usually do, but it can also be accessed via Python and other programs that touch the API. Um, all GDC services that are public facing are built on top of the GDC API. So if you're doing something on the portal, submission portal, exploration, and you think maybe there's something I should be able to do here, and this should be more flexible, that is likely something you can do through the GDC API. It's just when something's constrained to a user interface, it becomes naturally less flexible. There are outside researchers who develop third-party applications that use the GDC API as well. So this is all publicly facing, and we actually do have a page where we collect third-party applications that use the GDC API and link them through our website. It's called the Community Tools page. I'll mention it later in the presentation. Um, so the GDC API can be accessed directly using a multitude of endpoints depending on what you want to do. So here's the basic query structure of the GDC API. api.gdc.cancer.gov/ and then some endpoint. So as some basic examples here, files, cases, projects, endpoint give information about what you'd expect, files, cases, or projects respectively. But there are another set um, of endpoints that will query some of the analysis features or submission. So whatever you're trying to do with the GDC, you can probably do that through the API if the portal or UI features aren't satisfying. Now for my demo, I downloaded a, a chunk of open access math files. And if you're aware of those type of files, you'll know that they're actually pretty small. A lot of times when researchers want to download from the Genomic Data Commons, they will be downloading BAM files, which can be uh, hundreds of gigabytes, and this adds up a lot. Um, if you're familiar with how downloading via a browser works, you would know that this isn't a good idea to do through the browser. So the GDC has developed a standalone tool called the GDC Data Transfer Tool. And what this allows users to do is to download files via the command line by simply passing a manifest that comes from the portal for a specific set of files. And there are some nice features in here, including restarting an upload kind of where you, or sorry, restarting a download where you stopped if the download was interrupted. And it takes advantage of multi-threading download, so it can download many files at once, especially small ones. The GDC Data Transfer Tool is available for Windows, Mac, and Ubuntu. This is how they're available right out the box. But if you have another operating system and you wish to use this tool, contact the GDC Help Desk and we can help you build directly from source because it will usually work on most operating systems if it's built that way. Now I'm going to talk to you about GDC data exploration and how mutations can be browsed dynamically through the portal. So the GDC exploration page mostly features processed somatic variant data. This data is stored in the GDC API and also associated with clinical and biospecimen features um, directly through the case it is associated with. Um, the it's built more or less in the same format as the repository that I showed you before. So mutations in genes can be filtered using a faceted search, and custom sets can also be downloaded by building a cohort, and that can be saved on the website. There's lots of metrics and features that allow mutations and mutated genes to be viewed by their abundance and projects or across the GDC. So I didn't put it on here. There's a nice survival plot that will show you the survival for the cases associated with your cohort. There are summary plots that show which genes are most frequently mutated, as seen on the left here. And there are lollipop plots that demonstrate the position of each mutation on a gene along with the PFEM domains, which are just ways to annotate certain structural or functional areas on a polypeptide chain. As kind of a parallel section to the exploration page, we have the analysis tools. The analysis tools in the GDC are used to compare or visualize case, genes, or mutation sets. Um, and these case, gene, and mutation sets can be generated, like I mentioned before, in the exploration page and saved in in the GDC. So it'll be saved until you start a new browser section or until the GDC does a new data release. So there are options to export it to your local machine if you're afraid that might happen. So I'm going to show you a couple of these analysis tools. I'm not going to go over all of them, but first I want to talk about set operations. This allows two sets, so this can be mutations, genes, or cases. It allows them to be compared and their various intersections to be selected. It can be up to three sets, and subsets of these can be saved as new sets in the GDC. And you can take these sets and say, I want to browse for all the files associated with these cases, for example. Um, Down here, I kind of put one of my favorite use cases for this analysis tool, and that is I have two sets. And my goal here is I want to know which cases in the TCGA GBM project have both whole exome sequencing and RNA-seq data. So I would make two sets. One are cases with whole exome. The second set is cases with RNA-seq data, and I would run them in the set operations analysis tool, and that would give me this intersection, which is 161 cases that have whole exome and RNA-seq data, and those could be saved or viewed on another page here, as well as you could save the other sets, the whole exome sequencing data with no RNA-seq and the RNA-seq data with no exome sequencing, of which there are 5 here. So that has been useful for many users in the GDC. The clinical analysis tool allows for clinical data for a set of cases to be displayed using dynamic custom graphs. So one of the most customizable things in this are the bins. So bins can be customized according to their values and combined when necessary. Um, And then each clinical facet can be removed or added to the analysis. So for an example, if I wanted to make a graph about the race of a particular set of cases, and I didn't want to have two separate fields for the missing and not reported, um, I could either remove those or combine them into one unknown bin. This could be done for any of the clinical fields that are populated for a certain set of cases. The next thing I want to talk about is GDC data submission. And this is kind of how data first starts out in the GDC. Data comes in from external research groups, and that's where it goes through processing and then is made available in a lot of the previous tools I showed you before. So the GDC data submission portal is a UI that that can be used to upload metadata and guide data submitters on what needs to be done and what has already been uploaded. The submitted data that is uploaded to a particular project is browsable, and it's updated in real time. So if you submit 100 samples, as soon as those 100 samples validate, those are browsable directly through the submission portal. Um, Data harmonization can also be requested directly from the portal. Um, so one feature that's relatively new in the GDC data submission portal are the QC reports. This audits submitted data to verify that it is suitable for harmonization. And it gives you a report and shows which experimental strategies and which items are causing errors. And it blocks data from being harmonized if certain criteria are not met and these errors are not corrected. So for example, if read groups that belonged to RNA-seq, or at least were marked that way, were included in a whole genome sequencing read file, this would block harmonization until resolved because our bioinformatics pipelines wouldn't know what to do with this, and that would cause a lot of unnecessary trouble. So this blocks that sort of thing until this could be fixed, and then everything is unblocked and the data would be harmonized right after that. The GDC data dictionary is kind of the first line of defense for data quality. The GDC data dictionary gates submission based on required properties, so some things are required for certain items, values, so sometimes there are enumerated values, so only, for example, for the sample type, only certain fields can be put in there. So things like additional metastatic, primary tumor, blood-derived normal, that kind of thing. And data types. So some fields will only take things like integers. There are a few free text strings, and some will take only Boolean, true, false. This GDC dictionary is automatically implemented upon data submission. So if you try to upload something that breaks data submission or that doesn't follow the data dictionary rules, it will throw an error right away. Whereas the QC reports will give you an error a little bit later. So now that we're done with data submission, I'd like to talk to you a little bit about the GDC bioinformatic pipelines. This is how the GDC processes genomic data. So because GDC's purpose is to harmonize data, all alignments in GDC are performed on the exact same reference genome, which is GRCH38. If you go to the GDC website and you find the reference files, those are you can find the specific FASTA format version of the reference genome that is used in all of our analyses. So you can know exactly which one is being used. The alignments aren't performed with the same software, they're actually performed with different software based on experimental strategy. So for example, most DNA-seq data is processed using the BWA aligner, whereas the RNA-seq data is processed using STAR. So the next thing I want to talk about is whole exome and targeted sequencing processing. They're mostly the same pipeline. Um, so somatic variant calling after alignment is performed using 5 variant callers, and this is followed by annotation. These variant calls are available as VCF files, and these 5 variant callers are mentioned on the right here. Mutec2, Muse, Somatic Sniper, Veriscan2, and Pindel. The first 4 are for simple somatic variants, and Pindel is for small insertion deletion um, mutations. Um, so this is mostly done using tumor normal pairs of alignments, but there is some tumor only calling that happens. The maps are generated right now on an aliquot pair level per variant caller. Um, so in the past, if you remember GDC a few years ago, maps were available only on a project level. This was mostly for TCGA. Um, since we do have a lot of projects now that are ever evolving and adding things, maps are now generated on an aliquot pair level, and we um, we make an aggregation of all callers for that aliquot pair available. For those of you who are used to project level maps, the GDC developed the GDC math tool, and it automatically of aggregates a set of aliquot level maths into an aggregated math, whether that's project level or just cohort level. So that can be aggregated according to a manifest that it is given from the portal or by giving it a particular project, which it will then give you an aggregated math. Um, I do want to point out this webinar that was done in September 2020. It was really great and the bioinformatics team went very in depth for how the variant calling, VCFs, and math generation all happens. So I highly recommend looking into that webinar. The whole genome sequencing processing is a little bit different. It uses a pipeline that was developed by the Sanger Institute and does 3 types of variant calling. Simple somatic variant calling using Caveman and Pindel, structural variant calling using Brass, and copy number variant calling using ASCAT. And these are relatively new, and you will find them um, in a few projects, but that is growing as new data releases come out. The RNA-seq processing alignments are done with STAR, and for newer projects, 3 RNA-seq alignments are made available. Those are the genomic, which is the full genome, the transcriptomic, which are only the transcripts, and the chimeric, which focuses on reads that went across different chromosomes. The gene expression files that are made available from those projects are available as counts from both STAR and from HT-seq, and the HT-seq data is normalized via FPKM and FPKM upper quartile. Um, In addition to that, the splice junction files are also made available. In addition to gene expression, we do also have transcript fusion data available for some projects right now, and that transcript fusion data was generated using Star Fusion and Ariba. MicroRNA-seq processing um, is not as complicated as the ones I mentioned before. It uses the BCGSC microRNA profiling pipeline and makes, in addition to the alignment, the microRNA expression file is available for both full microRNAs and for isoforms. So those can all be filtered down using the faceted search on the portal. A relatively new processing pipeline that the GDC has is the single cell RNA seq processing pipeline, and that also will sometimes include single nuclei RNA-seq. This is a new data type for the GDC and is currently available only for the CPTAC3 project. The expression analysis files are available, and they can be used to generate t-SNE, UMAP, and PCA graphs. So first gene expression files come directly out of Cell Ranger as raw counts or by filtered counts, which reduces some of the noise. And then the Seurat package, which does the expression analysis or kind of a secondary analysis, gives the analysis file in TSV, which is the t-SNE, UMAP, and PCA plots, differential analysis, and the full analysis log in the HDF5 format. Some other miscellaneous experimental strategies that are mostly done for TCGA and a couple of other projects are first copy number variation. The TCGA copy number segment files are used are generated from the DNA copy R package, and those come from some legacy TCGA data. This data is also processed using GISTIC, which produces gene level copy number files on a project level for TCGA. There's also the methylation liftover pipeline, which is used to add information to methylation data that was previously produced by TCGA, and the coordinates were converted to GRCH38, specifically the version that the GDC uses. Lastly, there's the somatic variant liftover, in which variants were delivered directly to the GDC. So they weren't called, they were never BAM files at the GDC. They were just given directly to the GDC, and they were modified to have the HE38 coordinates that I mentioned before. These two types of liftover files are available for the Foundation Medicine Adult Dataset and the AACR Project Genie data that is on the GDC. Now I'd like to talk to you about some upcoming GDC updates. These are some changes that are coming in the near and long-term future. So first I want to mention that the GDC uses the GenCode version 22 data model, and we're updating it to version 36. The same exact reference genome will be used if you were wondering. This will replace any files that depend on gene coordinates. So these are things like RNA alignments, RNA expression, and variant calls. There are other files too that will be replaced, but this is just a subset of them. And this will be implemented as a large-scale data release. I encourage anyone who cares about this to join the GDC listserv. And if you join that, you will get a notification well in advance for when this will be happening. In addition to that, we will be doing some updates to the QC reports for clinical data. Now, the clinical QC reports I was showing you back in the submission section of this presentation focuses mostly on molecular data and harmonization. If the data doesn't look like this, it can't be harmonized. Um, but there are some more mistakes that can be made, most notably with clinical data. These QC tests will be implemented to verify that clinical data is suitable for release in the GDC portal. Um, Harmonization can happen beforehand, but when release happens, we want to make sure the clinical data is in good shape. An example of this would be any event that occurs before the days to birth date will produce an error because that doesn't make any sense. Um, in the in a future release, the GDC will release Jupyter notebooks that allow for specific analyses to be performed on GDC data. This release will happen via GitHub at first, but eventually Jupyter notebooks will be integrated into the GDC and will allow for seamless data exploration and processing with Python. The soonest release that I can talk about is the data release 29 that is upcoming. This will include browsable mutations for the Count Me In program and the CPTAC2 project. It will also include the MSI, the microsatellite instability status and value, being made available through the API and through the faceted search in the CPTAC program. And in addition to that, new cases will be made available for the HCMI program along with their associated files. And the last thing I want to really quick for anyone who's interested is go over GDC documentation. There are two main sites. The first is the GDC documentation website. This this is a user guide. This is documentation of features, how to use them, how specifically certain data types are processed, and we have it divided into the many features: API, data portal, data submission, etc. And all of our release notes are here too. So if you're wondering about any of that, docs.gdc.cancer.gov. The GDC website, which is found at gdc.cantor.gov, has higher level feature documentation and more of a general higher level um, overview of the GDC. This also houses the publication pages, which hold data that comes from publications that process GDC data, but the data didn't fit into the GDC data model. For example, some summary tables or something like that, or figures. These are just made available directly through the publication pages. And next, I want to talk about the community tools page, which I mentioned before. These are tools that were built on top of the GDC API and not developed specifically by the GDC development team. I can think of actually one exception, but some are generated internally by GDC affiliated people. All the webinar pages and the videos are made available here. So all of the webinar months that I showed in previous slides can be accessed through this website. And we also have an extensive frequently asked questions section that has some specific questions about most aspects of the GDC and some specific questions about the TCGA program. I would also like to plug our next month's webinar, which is going to be on using the GDC API. We're actually going to go over some examples of GDC API use using Jupyter Notebooks. And this will be on April 26, 2021 at 2 p.m. Eastern. That's the end of this presentation. Here are some useful links. And at this point, I guess we will take any questions.
Hi, Bill. There's several questions in the Q&A. The first is for future reference. Can we get a copy of Dr. Wysocki's slides on this GDC overview?
So this will be made available as a YouTube video and not a set of slides. So you will be able to see these slides on a YouTube video rather than a PowerPoint presentation.
And there's another similar question. Somebody went to the website and they couldn't find the video link. So do you want to bring up the support page and show the links?
For sure, yeah. Um, so let me go to Google Chrome here. So I'll show you a few things. gdc.cancer.gov. You'll click on support, and down here is monthly support webinar. So when you click on that, you'll see all of the webinars um, in reverse chronological order. So here's the GDC overview for today. Here's February's, January's, etc. Um, if they do have a YouTube video available, it'll be made available here. And you could also, I think there's a link in here to watch it on YouTube once you press the play button. The other thing I want to show that I was mentioning before was if you go to the support page, um, you can click on, sorry, you hover over support and you click on join the GDC user listserv, and this will bring you to a website that you can use to be on our notification list for data releases. If the GDC is going to be down for a small period of time, we'll mention it on there. And and for the Gen code update, you'll be made aware via this here.
Okay. What are the criteria based on which the new elements go into the API versus the supplement files?
Ooh, that's a good question. So it's kind of we take a look at how useful the data will be. So how useful is this data to somebody processing cancer data, somebody doing cancer research? Also, um, how populated is this data? If somebody is going to give us 1,000 cases, but only four of them have this specific field, it would be best to just keep those in the supplement file rather than keeping too many unnecessary properties in the dictionary. And yeah, those are kind of the main two criteria I can think of right off the bat. But we do have a really good presentation that we did on the GDC data model back in March 2020. We had our clinical data curator, Tara Lichtenberg, who gave a very good presentation on how data model changes were selected. So I would highly recommend checking that out.
Okay, and there's a question, can we search GDC using SRA identifiers and accessions?
SRA identifiers, usually not. I'm not too familiar with the way SRA works, but if they have some link to dbGaP, some of those dbGaP projects will be linked to GDC. So, for example, so in short, no direct link from SRA to GDC. But if you have a dbGaP number, you can link it to a specific project. So TCGA is PHS 000178.
Okay, the next question is, how frequently is dbGaP access checked?
I think right now it's around every 6 hours or so, but don't quote me on that. If you I would say contact the user services help desk, um, if you want a specific answer. I might have been wrong when I said that. Apologies.
Is there a containerized version of the GDC data transfer tool?
Sorry, was that containerized? You cut off for a second.
Yes, it was containerized version.
I don't believe there is quite yet, unless somebody developed it third party. But it's kind of an all-in-one tool. So It's just kind of one file. It's not a bunch of files in a folder, and it's a Python binary. So yeah, that's where we are right now.
Okay, and this user said, I tried to download some manifest files both from the cart and outside the cart. I'd like to ask for a suggestion why, by downloading a manifest file from the cart, I get validated data, while downloading the same file outside the cart, I get release data.
Oh, okay. So that might just be kind of an interface thing. Um, so either way, the data is released. If it shows up in the portal, it is released. I think one of the manifest functionalities gives you one state, and one of the other manifest functionalities gives you a different state that's used for a different thing. Both manifests should function the same exact way when you use the GDC data transfer tool.
Okay, I'm working with TCGA RNA-seq data. I've been using the GDC package and TCGA biolinks packages in R. Which one is more consistent? One file I'm interested in is a histological field. I'm interested in is the histological type that is only available through TCGA biolinks.
Oh, interesting. So my knowledge of TCGA biolinks is that it actually, in addition to grabbing data from the GDC API, it has a lot of external places where it knows to grab certain tables, certain metadata that aren't available readily in the GDC. So if you're using TCGA biolinks and you have any questions about where any data comes from, I'm sure the author of that package would be able to answer that for you.
Okay, and does the GDC repository have information about the area micro dissected from the histology slide available in the repository that corresponds to the molecular data?
Um, so for that, varying cases and projects have varying levels of biospecimen data richness. So if you were to go down to the biospecimen tree here, we have samples, portions, analytes, and aliquots. The portion is the part of the sample where the in which the analyte was extracted. Um, so this is most of the data that you can get from this. Um, Portion number one could potentially be tied back to some data that shows exactly where it was from the sample, but I'm not 100% sure where that data would be.
Okay, that's all the questions that I see in the Q&A. So I think we're just about done with today's webinar. Actually, I'm sorry, another question just came in. This is good. Is there an easy way for us to find matched normal samples for a data type of a specific tumor? For example, the matched RNA-seq data for people without skin cutaneous melanoma.
Yeah. So if you wanted to do that, one of the filters that you could add is the samples.sample type. I think actually if you just type sample type, it should be here. So what you could do is download specific normal or tumor samples from here, add them to your cart, and you could match them by case. Alternatively, something I'd like to show you is if let's find an RNA-seq data set right now. Um, I'm not going to find an RNA-seq data set because a lot of them in the GDC don't actually have matched normals. However, I'll download a whole exome sequencing data set. Let's say somatic sniper, BCF. and move everything from the cart. Oh. And we add all these files to the cart. What you could do is download the sample sheet. Now, what the sample sheet will show you is it's a TSV that will show you which cases are associated with each files and which sample type are also associated with those files. So if you were to take this TSV, you could process it using either, Microsoft Excel, Google Sheets, or if you're skilled with Bash, you could do that too. And you can look at which, this is a bad example because these have both tumor and normal in them, but you could see if they were only associated with one, you would see one patient associated with tumor or normal, and then you could filter, you could sort by patient, and then you would see this tumor and normal line up with each other. But like I said, this is a bad example because VCFs are usually matched tumor normal, as you can see they are appearing here.
Okay, here's a two-part question on clinical data mapping. Generally, what are the rules for selecting properties that are mapped to a given entity? Specifically, some attributes like BMI, height, weight were moved from exposure entity to follow-up. What was the rationale for this remapping? Our program collects and considers these baseline measures instead of follow-up.
Oh, okay. So they were remapped to the follow-up, but the idea there was it's only named follow-up. Here it can be anything that will change over time. So theoretically, you could put somebody's BMI and they could have a changing BMI associated with date values. However, many groups just put no date and they put the BMI into the follow-up. And that in that case, that would just be the same as putting it in the exposures. This is just a way we were doing that to make the data model a bit more flexible.
Okay, I think that's about it then. Bill, thanks so much for this, giving this webinar, and thanks everybody for attending. As Bill said, we'll get this out, the recorded video out on the GDC website as soon as we can. Thanks everybody. Have a good day.
Thank you, everyone.